Thanks to those of you guys who made it in real life, and thank you to those slowly trickling in on the Zoom. It's my distinct pleasure to be able to introduce the incredible Dr. Megan Gunnar today. She's a longtime champion and hero of mine, and she happens to be a distinguished McKnight University professor and a member of the University of Minnesota's Distinguished Academy of Teachers. She received her PhD in developmental psychology at Stanford University and subsequently completed a postdoctoral fellowship in psychoneuroendocrinology at Stanford Medical School. And she then joined the faculty at the University of Minnesota and has been there leading an incredible program ever since. Dr. Gunnar has spent her career studying how stress biology affects neurobiological and behavioral development and the processes that help children regulate stress hormones and subsequent functioning. She has many awards. I'll just name a few here. She's a fellow of the National Academy of Sciences, the American Academy of Arts and Sciences, and the National Academy of Education. She has lifetime achievement awards from many places, including the American Psychological Association, the Society for Research in Child Development, the Association for Psychological Science, and the International Society for Psychoneuroendocrinology. So she's hit all the big ones in her field and many outside her home field. She's really impressive in terms of her breadth. She also, of course, has mentoring awards. She is known for her incredible connection with trainees and junior faculty and has just been such an incredible power in so many people's careers that have launched to great success. She has awards from the APS as well as the International Society for Developmental Psychobiology, and I'm fortunate to count myself among one of those who has benefited from her incredible wisdom and insights. She's also involved in many activities to translate this research and make it applied, make it have real life value, and to help policymakers, practitioners, and families use this, including being a founding member of Harvard's National Scientific Council for the Developing Child. And finally, she serves on many important boards, trying to make a real world difference. And in particular, I'll just highlight that she is on the advisory council to the Children's Cabinet for the state of Minnesota, where that state is doing incredible things for families right now. And we are thrilled to have her here at UCSF and really excited to learn from her today. Thank you, Megan. Join me in welcoming her.
Well, it's lovely to be here. I feel like I'm teaching in Psych 101, where they actually broadcast it, so only like 5 people show up. No, this is very nice, but I'm glad you spread out, so I'll have people to look at in all different quadrants of the room. It's really nice to be here, and thank you for that lovely invitation or acknowledgement, Nikki. So what I'm going to talk to you about today is the calibration and potential recalibration of the mammalian stress system. And together, I hope we can think about what the implications might be, it's good to see you, Allie, for developmental psychopathology. Whoops, there we go. Now, no surprise to any of you, stress and the activity of stress biology is implicated in many different diseases, from everything from making it your colds and flu worse, or COVID, to issues of stomach ulcers, heart disease, and for what you all care about a lot, it plays a role in precipitating, amplifying, and maybe continuing depression, problems with post-traumatic stress disorder, and so on. Obviously, with this range of things, there are different mechanisms that are involved, and frequently what the biology of stress is doing is not necessarily precipitating or causing all of these, but amplifying and supporting the processes that lead to these problems. But as you also know, I assume, stressors affect some people more than others. Stressors are not equitably distributed in the population. There's a great deal of inequity, and stressors and adversity affect some people much more than others, typically those who are at the lower end of the economic spectrum or those who are minorities and experience discrimination. But in addition, some people are just simply more stress reactive than others. They activate more highly and for a more prolonged period of time. And these two issues are related, right? So your experiences of stress will influence how you react. How you react may influence how you navigate the world and interact with things that are going on in your environment. Okay, When I say the mammalian stress system, what am I talking about? For most of the work that is done with humans, because we can't be too invasive, especially the work done with kids, we are talking about the two major arms of the mammalian stress system. We're talking that both converge on our friendly adrenal glands.
Unexpected had happened.
Hello. What you're looking at there, these lovely things, are your kidneys, and these little hats that they wear, those are your adrenals. They have a cortex, which produces which is part of the hypothalamic pituitary adrenocortical system, produces cortisol in humans. They have an inner core, a medulla, which is directly connected with the nervous system and produces, is part of what we call the sympathetic adrenomedullary system, and it's what dumps adrenaline and is part of the fight-flight system. These are the two major arms. Both of them are regulated by the central nervous system. Both of them have effects on your brain. The adrenomedullary system produces a hormone that cannot get into your brain, but it interacts with the parasympathetic nervous system, causing feedback loops going up through the nucleus tractus solitarius to areas in your limbic systems, helping your body talk to your brain. But cortisol gets into the brain, gets into every cell in the brain. In fact, it gets into every cell in the body. And that's because two critical things, and I focus on cortisol, and most of the work that we're talking about actually does focus on cortisol for these reasons. It's a steroid hormone. That's important. Steroid hormones are lipid soluble. They go through cell membranes. They don't need active transport. If you take a bunch of cortisol, you're going to have it in every cell of your body before you know it. In the cells of your body, and in most cells of your body, there are receptors for this hormone, MRs or GRs. It's a whole long story. We're not going there. And once the cortisol gets into your cell, it interacts with its receptors. It causes the knocking off of some heat shock proteins and some things like that, activates those receptors. They're taken into the nucleus, where on all the genes that have a glucocorticoid, that's a fancy name for cortisol and corticosterone together, receptive elements, they attach and interact with those and regulate gene expression. There are many, many, many genes with glucocorticoid receptive elements. So if any, you guys know this, if you've ever had a need to take cortisone, it's huge. It's really a miracle drug. It's powerful effects all over your body. If you've had a cortisone shot in your knee, you know it's pretty powerful. Excuse me, good thing I have water. So it regulates gene expression in many, many cells in the body. Many systems are responsive. Many in the brain are responsive to glucocorticoids. And during development, it regulates genes that affect the systems that are developing. So it shapes, it influences brain development, which is why I've been so interested in the activity and regulation of this hormonal system throughout my career. Now, if we activate this system and activate it frequently, there are changes that occur. And Bruce McEwen was one of the ones to really emphasize this, give it a name. And when he was part of the MacArthur Network, they developed these ideas along with people here at UCSF. And they started talking about this concept of allostatic load. And that's the idea that if you this system's really important. You do not want to not activate the HPA axis when you need it. That's really not good. And that top there is an example of you need it, you activate it, you turn it off. But what they started talking about was if you frequently have to activate it, or if you have a trouble turning it off, and those two things sort of get related, then you see an alteration in the way the system functions. And because it's affecting other systems, you see alterations in the way other systems are functioning. And that is allostatic load, and it helps precipitate many of these diseases. What you are probably used to, unless you live and breathe this hormone, which I don't think, well, some of you probably do. What you're used to hearing is if you frequently activate the system, it goes hyperactive, and all of this bad cortisol that's over too much is causing all of the problem. And that is true. You can change the system and it can go hyperactive. but it also can go hypoactive. And both of those things are related to problems. Hypoactivity is related to a lot of chronic fatigue, pain syndromes, and other sorts of things. You need to be able to activate the system. Hyperactivity, well, I'm here to tell you, we actually don't see that much in people. Hold that thought. Because what we're going to do now is move and talk a little bit about sensitive periods for calibrating this stress system, which is a little different than the constant grind once we're not in those sensitive periods. And we know a lot about this, or we think we do, from more than 60 years of research on our friend, the rat, and then later, the mouse. And what we know from all of this amazing research is that if you either pick really good mommy rats or not so good mommy rats, hello, dams, you see a different pattern of stress activity in their offspring. And if you cross foster, you can switch that. Or if you muck with the nest in a way that will produce a disordered mommy rat, you will see the same thing. So poor nurturance, either low licking and grooming and archback nursing, what a mommy rat has to do, or disorganized. If you don't give her enough to make a good bed, she can't get her act together. And so she's a little of this, a little of that, and very hyperactive. And that's not so good for the rat either. And what you get is a rat or a mouse who is more fearful, who shows a larger and longer lasting corticosterone, that's rat cortisol, response, who gives up easier in like a Morris water maze, and is a poor, if they're female, poor mothers of the next generation, because all of the work by Michael Meany and others has shown us that part of this is carried forward through methylation of glucocorticoid receptive genes and estrogen receptive genes, and so on. So what a story. You have poor early nurturance producing a hyperreactive HPA axis, and you all are very familiar, I assume, in California because of your head medical director, with adverse childhood experiences. And this story, the big story with that is, boy, you have these experiences, you're going to have a really stress reactive kid who is going to be at risk. They're going to produce a lot of HPA axis functioning, activity, cortisol, etc. I like this version of adverse childhood experiences that was produced by Bruce Ellis, because it puts those experiences in their context, which is adverse childhood environments. Okay. And so on. So that's the story that you're hearing. And I'm here to tell you that it's actually a nuanced story, and it's a little different. And newsflash, we're not rats. When I started beginning to look, once I understood how we reacted and parents supported the regulation and all the rest of that, which was the first part of my career, when I started looking at kids who had really adverse early childhood experiences, I kept finding that they produced less cortisol in response to stressors. It was not what I was supposed to find. And I called up Gig Levine, who had been my mentor on the neuroendocrine side, and I said, Gig, the adrenal is not doing what it's supposed to. And he said, it is, and you just don't understand it, which was not very helpful, but it was absolutely true. We go on, I'm collecting data that is not what the data is supposed to look like, and so are other people. As we get salivary measures, we're able to actually get out there in the real world. We start looking at individuals who've had adverse experiences. And in 2017, this very nice meta-analysis was produced by Punia and colleagues, where they looked at a bunch of studies. There's your list of studies over there, of adverse childhood experiences and children and adolescents, but mostly adults, responding to social stressors, mostly the Trier social stress test. And what they found was either no effects, or you guys, I'm sure you're used to looking at meta-analysis. If you had produced a hyperreactive stress response based on these early experiences, all those dots would be over in this column, or really over here. No, they're here. Most studies either found no effects or hypo response. And the evidence suggested that was even clearer for individuals who had experienced maltreatment, and even clearer for the adults. So it emerged over time that they began, and so on. So we have a hypo-responsive system when nasty things happen to us when we're kids. And these were adverse childhood experiences, so it went all the way up to 18. So we don't know whether early, but in humans, usually if life isn't good when you're young, it continues to not be so good. It's somewhat rare, and I had to look for a group of kids like this where you were not very good, and then the world changed. Now, in 2005, I had a hint that I wasn't too far off, because up north here at your primate facility in Davis, Capitano and others were finding that when they looked at different cohorts of baby monkeys and they they assessed the adrenal, they found hypoactivity as well, and in a really interesting way. So basically, they studied 3 cohorts. This is the data from one of them. They all looked similar. These babies grew up either in a field station with a bunch of animals, sort of as normal as you can get in captivity. Either they had these corn cribs where they put the mom and a baby, or inside mom and baby, or they were hand-reared in the nursery. Animals were being reared for all these different experiments. And they looked, when the babies were 3 to 4 months of age, translation, about a year or so in human. They looked at their response to being separated and relocated, to the levels in the afternoon after that, to a dexamethasone suppression test, which is giving dexamethasone, which turns off the adrenal at the pituitary level. And some of you who deal with adults with depression might use a dex suppression test. There's an escape from that for depressed adults. And they looked at, and this is the really telling one, they injected ACTH, which is what the pituitary produces, and it directly stimulates the adrenal to produce cortisol. And what they found in every instance is these are the nursery guys. They're not responding as much. And the fact that you can directly stimulate that adrenal and it doesn't produce as much says some of this is not necessarily happening up here. It's happening down in the adrenal level of a down regulation. Why? Very possibly is protecting that developing organism, that it's what's nature's way. Okay, so is this what I would see in kids? And in looking for my model, I was invited to go to Romania, and we had the fall of Ceausescu, the opening up of orphanages in Russia and Eastern Europe, ultimately all sorts of children available for families who had been looking to adopt. And there was this real upsurge for about 10 years in adoptions, not from Romania, it only lasted 2.5 years. But from the rest of the world, China opened up. It was amazing. And this is an interesting model. They're also important children in their own right. But as a model of what I needed, we had kids who were experiencing significant deprivation, like the monkeys in the nursery, early in life. And then they get adopted into homes where parents want them, have looked for them. They're upper middle class to upper class. They're highly educated. In some sense, you can argue they go from some of the worst the world would offer to some of the most rich and enriching, not necessarily always the best, because we know there are a lot of issues for children who, especially children of color, who are being adopted into families who really don't understand how to raise a child who's going to experience discrimination. But for my adrenal question, at least initially, this was an interesting model. And when I went with Tony Earls and Maya Carlson initially to Romania, we collected data on 3-year-olds who were living in an orphanage in Iași, Romania. There were 46 of them. We had a group of kids in Romania who were at a daycare center, so we were able to work with their families and collect. This is a normal daily rhythm. You wake up, your levels are high, they're stimulating you to want to eat carbohydrates and explore the world. Then they come down and they go down to nothing around bedtime. And that's important for resetting the clock. Not one of these 46 kids had a normal rhythm. And the biggest effect was this low level in the morning and relatively flat over the day. Now, I came back, I said, well, that's really interesting. and came home, and I called my program officer at an IMH. I called them parole officers. And I said, wow, I really want to study kids adopted internationally, because I think this is from orphanages, because I think this is a way to really get at what's the effects of early experience and understand how early we need to go to do interventions. And the response of that program officer was correct. She said, well, do you have any of these kids in Minnesota? What's your sampling frame? And do they have any problems that NIMH would care about? So the first thing we did actually was a dog and pony show all around Minnesota to talk to parent groups, et cetera. It turns out Minnesota had a lot. We have a very big children's home society adoption agency that had been working with Korea for a long time, and began working with these others. And we then developed a survey, worked with DHS, blah, 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 fielded it in 2001 to about 5,000 on 5,000 kids to almost that many families, got about 4,000 in return. They all joined a registry. We kept doing that. I now have a registry of over 8,000 kids adopted internationally from orphanages and other conditions. But most of those kids now are adults, so I'd have to re-up them if I wanted to keep following them. And they don't just come from Romania, they come from everywhere around the world. In our initial pilot work that we did while we were doing the dog and pony show to get a sense of what we would see, we did see, boy, 2 months after you get to your families, you actually do have a diurnal rhythm, but it's not as high. So these are typically developing kids, here comparison kids, and these were our kids adopted from orphanages. So there were only a few of them. It's a pilot study. At that time, I was also running a network supported by NIMH, where we were trying to understand the development of the HPA axis. And I knew I was going to need a way of doing some random assignment, which you do not do. You do not randomly assign kids to adversity, but you might randomly assign them to improved care. And I had two people that I invited into that network who were doing randomized controlled trials of improving conditions for children who are at risk for foster care or in foster care. Mary Dozier was one of them, and Phil Fisher, just now down the road from you all at Stanford, was the other one. And Phil went and collected, I love this time, I had a little bit of money and I could just hand it out. And I gave Phil some money, and they went out and collected morning and evening cortisol on many of the kids that they were studying in their foster care intervention. And what they found when the kids first got into the intervention was, compared to typically developing kids, we calculated the mean and standard deviations based on my data on kids who had not been maltreated. Only about 4% of the kids had elevated morning. 62 had sort of like my typically developing kids, but 34% were more than a standard deviation below typically. They were hypo. So this led me to believe that I was not just looking at orphanage conditions, though it was interesting that when you did the analysis of child protection records, what predicted it was severity of neglect. Okay, so kids need responsive parents in order to activate that normal rhythm. But hypocortisolism is not just low court. So I needed to get beyond just, I was so hesitant to stress kids, right? So we just did a lot of normal collection as they went around. But finally I said, I've got to stress them because I need to know whether they're going to activate. And then I realized I probably was stressing them because we were doing a study of transition into the family. We brought the kids in. We saw them over a course of 2.5 hours. They were little, they were 18 to 36 months, and then every 6 months after that. That's a long time. And about halfway through, we slapped electrodes on them so we could get pep and vagal tone while they watched a movie. And we were also taking court. And so we took a look at what we would see over the course of that. This is session 1. Everything is novel because they're 18 to 36 months of age. These are my non-adopted comparison kids. Okay, oh my gosh, those electrodes in the second hour we went up. These are my PI kids. No elevation. And none. And we actually saw them every 6 months over the course of 2 years, I could show you the whole thing. These guys, with each time, responded less. They were getting used to the whole context. They were adapting. No change for these guys. None. Okay. All right. Sounds hypo. We created a score for hypoactivity. We made some predictions with it. I'll show those to you in a few moments. Where am I at? Yucky, too much. But we needed to move to like the real deal. And the real deal in human research is the Trier social stress test. It was developed by Clemens Kirschbaum. It was his dissertation in Trier, Germany, hence the name. And it was basically, it's public speaking. You prepare for 5 minutes, you give a speech in front of an audience while being filmed and told you're being judged. And then you do mental arithmetic, which in adults is like you start with, I don't know, 17,000 and subtract by 700 as fast as you can. And every time you make a mistake, someone says wrong, start over, okay? Public speaking is a social evaluative stressor. And that, since we can't point guns at people in our labs, which would be a threat to your physical self, we can do the social evaluation. Human beings are very sensitive to being socially evaluated and potentially rejected from the group. It is, if you think about it, because of our needs to stay in our social group and have a standing in them. Social evaluation is, and being found wanting. What's that movie? You've Been Tried, and the guy is the, pretending to be the swordsman, whatever. Anyway, and you've been found wanting. The Princess Bride, is that it? Yeah, exactly. And you've been found wanting, and you're out of here. Okay, so the Trier social stress test, which has now been adapted for kids. You prepare a speech, pretend you're introducing yourself to a new class. This is Jodie Quass's lead-in that works very well because it works for children and adults. Tell your class some things that they will like about you and some of the things that they might not like about you. So really emphasizing social evaluation of you. And then you do the math and you make mistakes and you're filmed and they're judges and all the rest of it. Now, we had started in 2013 taking a look at 9 and 10-year-olds. I figured they were old enough they could run away and they actually could give their consent, looking at their responses to this task. If they were non-adopted, that is, born and raised in their upper middle classes families in Minnesota, or adopted from institutions. This is actually a change. This is 20 minutes after you start the task, which is where you should be at your peak response, peak levels. If you look at my non-adopted kids, they show a response and the test is all over, right? And now they just return to baseline or a little below. These are my PI kids. They're flat and they just crash well below baseline. This is what we see. So in a context where you actually need to produce this hormone to sustain the energy in your brain and your thinking, you crash. All right. I don't get to randomly assign kids to orphanages or to adoption. It's a problem. However, the Three Musketeers, some of you know these guys, Nathan Fox, Chuck Nelson, and Charlie Zena, were part of this MacArthur network that did this incredible study in Bucharest, Romania, which is why it's called the Bucharest Early Intervention Project. They went into orphanages in Bucharest. They examined the kids to identify ones that had prenatal congenital issues, put those aside, and then they took the ones who they created, because Romania had no foster care. So Charlie, who has developed excellent foster care in New Orleans, created a foster care system for this study in Bucharest, and they randomly assigned, after they had done assessments, children to care as usual, whatever the Romanian government wanted to do with them, which was stay in the orphanage, and then things started to change, or to go into these foster families, which were supported. So this is the only study we ever will have on random assignment out of an orphanage. And this study ran until the kids were 5. At 5, all the children reverted to care of the Romanian government, which by then had realized that these children were not just intellectually inferior, which is what you see. Kids in orphanages go from looking basically normal to being about a year and a half delayed developmentally. So they look like they maybe have intellectual problems. The ones who went into the foster care developed intellectually. So anyway, Romania developed their own foster care system, which was terrible because they had gotten rid of social work. Anyway, long story. But now they had no more money from MacArthur, but they got money from NIMH to follow the kids. And when the kids were 12, they ran them through the Trier social stress test. And what they found, they also had a group of kids who were never institutionalized, who were born and raised in Romania. And those kids, so they brought them in, they played with a kid, they went through the Trier, they did a frustration task afterwards, and those are those kids. That just looks normal. These are the kids who were in foster care. These are the kids in care as usual. There we have our lack of a response again, but under random assignment conditions. Now, the way they set this up, the median age of the kids at assignment was 2. So they looked at the kids who had been assigned to foster care before 2 and after 2. This was really, they came in at the same time and the kids were a range of age. And these are the kids who got to foster care before age 2. These are the ones after age 2. My own work suggests that this may be an 18 to 24 month issue, but it really is looking like we do have a sensitive period for calibrating the system. And under conditions of deprivation and lack of nurturance and poor care, we go hypo. And we may well go hypo at the adrenal level, and I'll show you more evidence that that's what it is, not necessarily up in your head, which is why these kids look frightened and anxious and everything else, but maybe down low. Okay, and that's what I've tried to say. Does it matter for psychopathology? If I take a look at the kids who are really the most hypocortisolemic, and this was in our transition into the family study, we had kids from the orphanages and kids who are our comparison kids. I looked at their cortisol over the 4 times that I saw them every 6 months in the first 2 years and calculated the degree of hypocortisolism. We observed them to see the extent to which they engaged in what you may know as indiscriminate friendliness, or now it's got a new name, disorders of social engagement, which basically they treat people they've never seen as if they're intimates. And the biggest difference we saw at this age was they touched you. Like, you can have a friendly 2.5-year-old who might talk to you, but they do not climb in your lap. And some of these, so the more physical engagement with you, even though they've never seen you before. Then at the last session, we did Charlie's interview with the parents to assess this inhibited, this DSE. And in kindergarten, we observed all sorts of behaviors, their social skills, their victimization, their aggression, and behavior regulation. What we found is the cascade that you're seeing here. And so I think it does matter. The more hypoactive the kids are, the more trouble they're having with peers, the more trouble they're having with attention regulation, etc. Whether the hypocortical is causing that, or it's an index of how bad it was in the institution, which is what is causing it, I can't tell you because I can't. Okay, back to the rat. Really nice early period. Their period lasts, of infancy lasts a little less than 3 weeks. When all of this gets set, really it's in the first 2 weeks, and they're set for life. But are we? Rats live 2 years. And I mean, they are ready to go to reproduce within a very small amount of time, which is why never let your kid tell you that they want to have 2 rats, 1 male and 1 female, because you will have way more than that very soon. In fact, you'll have a whole, I mean, and then they'll say, oh, and then we can sell them because they're blue whatevers. Nobody wants to buy their rats. I had so many at my house. Oh my gosh. At any rate, they live for 2 years. The chance that the conditions that you were reared under will be the conditions in your environment over the course of your reproductive period? Probably pretty high. If you think about feast and famine periods, which is probably one of the biggest impacts for humans and animals, you know, they come and they go. But, you know, 2 years would probably set you up pretty well. In a human, you're just 2 years old. You've just gotten done setting it up. We live so long. We aren't reproductively fit until our teenage years. And in conditions sort of subsistence living, we're not really reproductively fit until we're around 18 or 19. Now it's earlier because we've got, you know, so many resources. So does it make sense that we calibrate our stress system for life in the first 2 years, and it's going to be that way till we're 90? Is it going to be that way when we're ready to give birth? Seemed a bit unlikely. And along comes our rescue. It's called puberty. Rats don't really have a puberty. They have a peripubertal period, but it is not in the same way as ours, where you get this massive flexibility and change in neural and in neuroendocrine plasticity. It is really a very plastic time. And it has implications for both our stress and our emotion systems. And you all probably realize there's a lot of interest in studying the plasticity and the impact of experiences during the adolescent period, with a very strong sense and some evidence from the rodent that if adverse things happen then as opposed to a little earlier in childhood, you may see a bigger impact. We had data already taking a look at age when we started using the Trier social stress test. It is easy to do things that wipe out the response, especially in Americans. Europeans, and Germans, oh, they respond to authority. But in America, not so much, which actually Clemens says is because our lives are so stressful, we've all gone hypo. But we could talk about that later. So when we first started, we were not really good at getting the youngest ones to activate. But that was great because this is what it showed us. It showed us that when I had 11 and 13-year-olds who were prepubertal, I was not getting much of anything. But with puberty, I was beginning to get a really nice response. So that's the evidence. And we have, that's a tiny piece of evidence. We have lots more that the HPA axis begins to be more reactive and produce higher levels of glucocorticoids during puberty, along with all the other crazy hormones you're producing, which are probably also changing the receptors for glucocorticoids, so you sort of activate and up the system. Okay? And that led me to think about, is this a period when we can recalibrate the system? And my ideas were, sensitive periods cut both ways. So if you started out under harsh conditions and went hypo, what would happen if your conditions were benign during this pubertal period? The alternative, which I was not able to look at, is what if you started out with benign conditions and the world caved for you? Unfortunately, we probably have plenty of kids in war zones where that question could be addressed, but I didn't have my access to them. So the idea of we have more plasticity here and more plasticity, and maybe that's what's happening. And therefore, I wrote and got from NIMH, NICHD, sorry. No psychopathology here, so they didn't get a study to see if we got some recalibration in these kids. The average age of adoption was 15 months, which is classic. The kids ranged in age up to 5 years, but the median age of adoption was early. 90% had been adopted by age 3. Parent education and income was similar in the adoptive and the non-adoptive. It was an accelerated longitudinal design. And what that means is I picked the kids up when they were 7 and 8, or 9 or 10, or 11, 12, 13 or 14. And then I studied them. And a year later, I studied them. And a year later, I studied them. And using fancy statistics, we're able then to piece together 9 to almost 17 years, the range. I was interested not in the kids' perception of where they are in puberty. Kids tend to normalize compared to everybody else, but they're real. So we did a nurse's exam to get puberty, and I measured cortisol, DHEA. It's an adrenal androgen that increases with puberty, but also reacts, correlates typically with cortisol. And I measured salivary alpha amylase to look at the more sympathetic side of things. Forget it, there's nothing going on there. End of that story. And one of the things I had to worry about is, were the kids who were adopted from orphanages advanced in puberty? Did I need to do the timing differently? And the answer is that story's way overblown when you actually do the right work and don't ask parents to respond to a questionnaire if they think their kid is advanced. And one of the first times in my life, the data came out beautifully. I couldn't have called Gig. He passed away, but the adrenal was doing exactly what it was supposed to, because here's what I saw. These are the non-adopted kids. This is actually the data looking across the kids. So I'm getting within child change here. And this is stages of puberty. So the earliest stages and so on up to, I had a lot more kids who had reached stage 5. We might have seen that they react more, but this is basically what we saw. No significant change really with pubertal stage here. These are my stage 1 kids from orphanages. Just what I showed you before, with a different set of kids. We replicated. And then as puberty increased, they began to respond just like the kids who were non-adopted. So we got a classic, beautiful recalibration. And these kids, remember, they were adopted at 15 months on average. They are 7 or older here. So for a long period of time, until puberty opened the window, they stayed hypo. There was something about puberty sort of opening that period, like the body then began to resample the environment. Sounds great, doesn't it? Okay, where is it happening? We had DHEA along with it. You can get that from saliva too. It increases with puberty. It's typically correlated with cortisol. Looking across these kids, we did not see differences in DHEA levels between and in both groups. DHEA increased the same way with pubertal stage. So DHEA produced in the adrenal is acting totally normally. DHEA, you start with cholesterol, you go through different enzymatic paths to get to DHEA or to cortisol. It's acting normal, but it didn't correlate with cortisol initially. So here's what you see. These are non-adopted kids. This is levels of DHEA going up with puberty. This is the degree of correlation with the cortisol response. No difference there, always correlated. It just increases the things they found because it's responding to ACTH. These are my PI kids. No association. It's going up just the same. No association, and then with pubertal development, the system gets organized. And I think this suggests that the enzymes are coming back in the pathway that responds to ACTH. So along with DHEA, now I'm getting the right amount of cortisol being produced. Does that make sense? I think it's happening it's happening in the adrenal. Well, okay, what is going on then? I've got a brain that's not used to seeing cortisol. I have an adrenal that now with puberty and a different kind of context is now probably changing its enzymes, so it's reacting appropriately to ACTH. And now a brain that's going to start seeing more cortisol. Does this mean the kids are going to look great? Are we recalibrating and everything's going to be sweetness and light? Or have we, in a sense, created a challenge for these kids? Nature created a challenge. One thing that we know for sure about PI kids is that adolescence is a more difficult ride than it is for non-adopted kids. Adolescence is a difficult ride for every kid, but it's a much more difficult ride for kids who come out of orphanages. And one of the sets of data here that we have is this beautiful data out of the English and Romanian adoption study, which was done by Michael Rudder. And since they were only looking at the kids who met clinical cutoff, I've been looking at symptoms, they look at cutoff. They didn't see much of a difference in emotional problems at age 6. But man, oh Manischewitz, by the time the kids were age 11, they were seeing it, and it got worse as they moved on into adulthood. Some of that was because they weren't able to meet the challenges of adolescence, but there was other stuff going on as well. So we asked, is behavior recalibrating too? And we had the health and behavior questionnaire that MacArthur had put together, the MacArthur group had put together with Tom Boyce sort of leading that when they had their little MacArthur network. And we had used that and collected and we looked at cross-panel analyses. We found no effect of any change in cortisol levels for my non-adopted kids, but as the PI kids increased in their cortisol production to the TRIR, we saw an increase in internalizing issues. Not a whole ton, but some. I have wonderful students, Nicole Perry being one of them, who is now at Texas in Austin. And they wanted to follow these kids up, give them more time. They weren't that deep into adolescence. What happens? Was this just a transient thing, or would we see longer lasting effects? So they followed the kids up 4 years after the end of our study. They were 13 to 21 years at the time they managed to get data out of them. Yes, this was during the COVID pandemic. So they also looked at issues of that. Parents and kids did questionnaires. We got a 68% attention from our last session. And it was really Nicole Perry and my lead research person, Bonnie Donzella, who did this study. Now, let me explain how this operates here. They looked at internalizing reports, parent and kid, externalizing report by the parent, because the kid never thinks they're externalizing, emotion regulation issues, and negative lability. They controlled for state anxiety because it's a pandemic and from day to day, Lord knows. Age, sex, pattern means did you recalibrate, did you show increases or not? Group is PI or NA. And they got a very significant group by pattern interaction. Didn't matter for the NA kids. For the PI kids, if they were recalibrating, they were showing more internalizing, more externalizing, problems with emotion regulation, and greater negativity, even later on. So I think the challenge we have to think about is you guys probably don't deal with many kids who go from wretched to fabulous. But if this is operating, the more aversive it is in puberty, the more recalibration, even more disorder you should expect. I think what I'm seeing with my kids is their brains were used to driving a Volkswagen and they've now got a Ferrari underneath them. They're producing that stress response. Will they ultimately figure out how to manage it? Is there something we could do that would help them? Is there something going on in the brain that we could interfere with? That poor amygdala is probably saying, oh my gosh, I never saw so much of this stuff. Or, you know, what would we do what would you guys do? I'm not in to try to work with kids like this. Should we be getting in before puberty? And I think that's the message for all of us, to really work hard on emotion regulation, so they're not putting themselves in stressor paths as much, stabilize their lives. And that's my talk. Thank you very much. I think we get questions.
She's a great storyteller and incredible scientist. We have time for questions.
Hi, thank you. Thanks for a wonderful talk. I'm wondering, so first, who ends up in orphanages, and has there been an opportunity with the recent explosion of large-scale genetic studies to be able to begin to look at the mediation of genetics in this progression that you see, particularly that last step between low cortisol and the emergence of psychopathology?
Yeah. So who ends up the answer to who ends up in orphanages depends on where in the world you are. Certainly you don't end up in an orphanage if you're coming from an upper middle class, blah, 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 blah. If you are in China, though, you end up in an orphanage if you have chromosomal problem, that is 2 X's. And so actually the little girls from China actually look better than the kids we see from Russia and Eastern Europe, where, for 2 reasons. One, I'm not sure they're exposed to as much alcohol prenatally, probably not. And in Russia and Eastern Europe, really, it's only been in the last 10 years, there have been campaigns against drinking during pregnancy. So we also assess facial features of fetal alcohol stuff. The issue of hypo versus hyper has not, to my knowledge, truly been looked at well genetically. You wouldn't need to go to an orphanage for this, because we see this you see this pattern in people who are exposed to traumas, even those who don't develop PTSD symptoms. So the classic is, like I said, humans downregulate, but some don't. And I've actually thought that that's going to tell us a bit about who becomes depressed versus other things. But I could be wrong. I'm not a clinician. There have been some studies, there were initial things, a little bit that Meta did, and so on with the Romanian, English in a Romanian study, looking at our favorite friends, BRD4, etc., and showing that, yes, if you carried those variants, you were more likely to develop ADHD if you were in an orphanage or not. So, I mean, it was an added vulnerability. But in terms of up and down regulation of the HPA axis, I don't know. I don't know that, and it would be a great thing to do to see who's got those genes. And quite frankly, from the data I'm looking at, I think I'd look at genes that are regulating, that are at the adrenal level and regulating those adrenal enzymes. There have been, FKBP5, though, is the one where we do have, and that's the real interest. That's, for those of you who don't know, that's one of the rate limiting genes for, or enzymes, proteins, that limits when you get cortisol into the cell, you have to bump that off in order to get it into the nucleus. And stress changes the amount of that product. It's one of the ways of the body to keep you from seeing too much cortisol. It will up or down regulate that. And there is all sorts of beautiful work on the genetics of that in relation to PTSD.
While we're bringing that to Anne, can you comment briefly on the word dysregulated?
Oh, we love that word, don't we, in psychology and psychiatry. We love to say if it's not doing what everybody else is doing, it's dysregulated. And I think the challenge for us is, and now I'm going to sound like, you know, Bruce Ellis and others, if this is what the pattern that we typically see in response to X condition, it's been selected for. So it's not probably a dysregulation, it's an altered pattern of regulation. That doesn't mean, though, that that altered pattern can't mess up other systems, because it all comes, anytime you do a change like that, there's a cost to it. And I think that's what Bruce was talking about with allostatic load. It's, yes, what's supposed to happen was selected for, but it has a cost. Yeah.
Thank you so much. So first of all, echoing also what Matt said about the importance
Could you talk a little louder?
Oh, sure.
You're on. It's just louder.
Okay. So echoing what Matt said about the importance of considering genetic, intrauterine exposure, and postnatal environmental interplays. During your talk, I found myself thinking a lot about stress in the medical profession. It's on People magazine, I think, last week. I don't read it, but somebody sent it to me. And thinking about allostatic load in adults. So what's interesting about physicians, and plus I don't know as much about other healthcare professionals, but I trained in environments that were kind of high stress, and so observed the personalities of people around me and had this idea that more neurotic people were going into medicine, and then that influenced our response to stress. But the data does not show that. So what's really interesting is that at entry into medical school, if we think it's a proxy of stress exposure, the rate of psychopathology of people who are going into medicine is actually incredibly approximative of general population data. That changes very rapidly by the first year of medical school. And so really thinking about, obviously, the incredible importance of sensitive period and how it impacts brain development, possibly in the context of other vulnerabilities that we need to map out, but also sitting with that idea that some of the things that you're talking about are obviously like they're pseudo experimental designs, like medical training or being a healthcare worker, that may be a proxy for very high allostatic load.
Yes.
And wondering what your ideas are about that.
Well, medical students have been the go-to for studying stress and immune. All of the early work on how the immune system goes awry was done on medical students studying for their exams, because they, of course, a captive audience. So what you say is absolutely true. Medical school, especially when we, decided we'd really bollocks up people's circadian rhythms by making them follow patients for 36 hours, and which was probably good for the patient, but not good for the medical student, is true. And that's when the source of studies of psychoimmunology for a long time and taking glucocorticoids. Yes, we will change our systems at any age. It's just during sensitive periods, it's easier to do it, right? So, put somebody through how many ever years of medical training, stressful. So yes, we all had really, you need to do stress reduction work with anybody under those high stress conditions. Yeah. Mm-hmm. Yep. So it looks like human beings tend to go hypo whenever. That seems to be more of our pattern, I think, than hyper. It may be because we're not lunch for many species. In fact, we have to live with those entities. We're only lunch if we leave the group, and the group is usually causing our stress. So we may have evolved to be able to keep that system at a lower level than the little ratty or mouse who's got to run or whatever. That's my theory. Don't hold me to it. So we tend to go hypo. The sensitive periods where I think it's easier to regulate that are periods of rapid change in endocrine systems. Babyhood, puberty, and I have a wonderful student, Marian Howland, who is really interested in pregnancy as another period with lots of hormonal changes, which may be a sensitive period, and might also be a sensitive period for being able to work with a woman psychologically, because especially with that first pregnancy, she doesn't know what end is up, and therefore, there's a lot of flexibility. One would also assume that menopause might be another one of those. So, those are interesting possibilities. Yes, two guys over there. We got two over here. And there's individual differences in what level, and blah, blah, blah is all the genetic stuff, probably. Last question. Fight over it.
Hi, thank you so much for this talk. Speaking of Volkswagen, kind of Ferrari, this idea, I know that with the recalibration, it sounds like the cortisol does increase during puberty. I guess I'm wondering, at the signaling level, having not seen cortisol for such a long time, in glia or neurons, are the receptors still expressed at the same level?
I need a rat person. This is not a question I can answer in humans. So we need a model, and the model is not going to be the rat, but there are some smaller rodents that might, more that we can work with. You don't want to work with a non-human primate on that, because we can't sacrifice them either. So we need a model to really figure out what's going on at the receptor level, unless you're really more creative than I am. And there are things we can possibly do with adult humans using, agonists and so on to maybe answer that question. One answer I do have for you is, interestingly enough, it doesn't look like they can't turn it off. So that suggests that at least at the endocannabinoid level, at the level of the hypothalamus, they're able to recognize it and shut down the system in the same way. But that would be endocannabinoids. It'd be the fast stuff.
I guess I'm wondering, as a proxy, is it possible to obtain post-mortem tissues, things like this, from adolescents who unfortunately get in car crashes and things like this? And so
you could begin to do that, but you'd have to have the kids. Tricky, but potentially worth it. We're developing more brain banks where you might be able to do that.
So hopefully you have a little better sense of some of your patients and the paths they've walked and some really good ideas for next studies, and also have just a tiny window into this brilliant mind who has just influenced our field in so many ways. Please join me in thanking Megan Gunnar for coming to be with us today.